profile alignment. So we learned about that, and that is the most used method for multiple sequence alignment. So once you have performed multiple sequence alignment, you can use that for making a tree, phylogenetic tree. And what phylogenetic tree does, what information it provides? Evolutionary, yeah, evolutionary relationships among organisms. Okay, yeah, distances between sequences, and based on that, you infer the relationships among organisms that have those sequences, biomolecular sequences. So let's get going with molecular evolution today. And here is a cartoon of 5-kingdom system: Monera, protists, fungi, plants, and animals. Okay. Now we have domains of life. So 3 domains of life, and they are bacteria, archaea, and eukarya. And this was based on 16S tree. Yeah, 16S ribosomal RNA tree. And also many different classifications or sub-classifications. For example, protozoa, invertebrates, vertebrates, and mammals. Okay, I'm not going to define all these. So you understand what is a plant, okay, what is an animal. So, but there are some characteristics that define these groups. So you would want to know those characteristics. For example, mammals. So any characteristic you can think of? Mammals. Oh, mammary glands, yeah. So, but there are multiple characteristics that can define a group of organisms. So these are different taxa, and this helps in understanding not just relationships among organisms, but also the shared structures and functions among these organisms. So that's important for many purposes. Yes.
You talk about 16S.
16S ribosomal RNA sequence. So you have not heard about that?
For eukaryotic,
I'm sorry?
that is used for prokaryotes.
Yes, prokaryotes only. So yeah, that's okay. You can, yeah, you can say just ribosomal RNA sequence, and that's fine. Okay. So ribosomal RNA sequence is a marker gene. It has components that evolve slowly, as well as components that evolve fast, right? So just like a clock. So uh, so all those needles for second, minute, hour. So it has been used for classifying organisms, prokaryotes and eukaryotes, and now we know that there are 3 domains of life based on that. I think that was 1970 something, 75 or 76, when Carl Weiss and Fox, yeah, I'm forgetting his full name. I met him in Houston. They developed a method for classifying organisms. Yeah, Dr. Weiss is no longer living, but Dr. Fox is, I think he's also retired from Houston, but I happened to meet with him when I visited Houston. So, so yeah, people have been actually classifying organisms based on so many different characteristics, complex morphological features, or sequences of nucleic acids or proteins. So ribosomal RNA is nucleic acid sequence. So based on that, different types of classifications. So the father of evolution, Charles Darwin, he published a book in 1859, and the name of the book is The Origin of a Species by Means of Natural Selection, or the Preservation of Favored Races in the Struggle for Life. This book introduced the theory of evolution. Have you anyone read this book? You have read it? Oh, you have. Oh, you have not read it. Okay. So yeah, I think very interesting book. It's not quite voluminous. It's actually a thin book, and I happen to get this book from Oxford University Press as a gift. And I think very fascinating stories there. So at some point you would want to. It was published in 1859. That's a long time ago. There is just one actually figure in the book. so, mainly words actually text. So individuals that are more fit for a given environment, they are selected for in the course of evolution. So individuals that are adapting better in a certain condition or environment, they are selected for because they can adapt better to that environment. So they are fitter to that environment, they will produce fitter progenies. So they will be selected in the course of evolution. So over longer time periods, because of this process, new species will emerge and evolve. Okay. Okay. So at the molecular level, evolution is a process of mutation with selection. So changes happen in biomolecules, for example, DNA and proteins. So changes will happen in nucleic acids. And then some of the changes will be selected for, some would be selected against. Okay. So, and that is selection is there for helping organism. So deleterious changes or mutations, they are selected against because those changes may result in disruption of the proteins and organisms may die. So over the course of the time or evolution, those changes are selected against, and changes that can confer novel traits to organisms can make organisms fitter in a certain environment. They may get selected for those mutations or substitutions. So, and over the course of the time, because of these changes happening, new genes can emerge, and because of this, new species can emerge. So molecular evolution is now, earlier evolution is based on many years ago complex morphological features. Now evolution is mainly molecular evolution. So it's a field of study that infer relationships based on the changes in DNA and protein sequences. Okay. And phylogeny, of course, is the field of science that deals with inference of evolutionary relationships. Okay, so the studies of molecular evolution began in 1950s, and you remember some developments happened in 1950s. What were those developments in the field of molecular biology or genetics? DNA? Yeah, DNA structure, 1953. So other than that, there were some more important scientific breakthroughs that happened in 1950s.
Sanger sequence.
Okay, yeah, Sanger sequencing, protein sequencing. So sequencing of protein molecules, and the first protein that was sequenced was insulin. And since then, dozens or hundreds of protein molecules from different organisms were sequenced. So it provided opportunity to compare those sequences to understand the difference between the sequences arising from different organisms and how these have evolved. And can organismal relationship be inferred based on the changes that were observed in these sequences? Okay. So Frederick Sanger, he sequenced insulin, and let's look at the structure of insulin. So insulin has signal peptide, so regions actually in insulin, signal peptide, beta chain, C peptide, and A chain. So the signal peptide and C peptide, they are cleaved out from the mature insulin protein. So what remains is the heterodimer of A chain and B chain. Okay. So because of that, because they are not needed actually for the mature protein, so there is less functional constraints on signal peptide and C peptide. On the other hand, there are more constraints on A chain and B chain. So you can see the mutations in signal peptide and C peptide. So there are many gaps, many mismatches, while B chain and A chain, they are highly conserved because of their functional significance. Okay. Okay. So, and also in the loop region, again, owing to the functional constraints, so less constraints in the loop region, resulting in more mismatches that you will see here. Okay, so and further, we came to know that signal peptide and C peptide evolves 10 times faster than B chain and A chain. Okay, the rate of evolution. So, number of nucleotide substitution per site per year. So, this is the rate of evolution. Okay. Okay, so surprisingly, insulin from guinea pig and from the related coipu evolved 7 times faster than insulin from other species. Why? So you would wonder that it is likely that these insulins have less functional constraints. So any thought of why? Okay, so you have not taken a look at the slides before coming here. So yes.
Due to less selection process.
Okay, so less. So yeah, this is what I'm saying. Less functional constraints. Uh, so what those constraints are on insulin from human, and they are not there in guinea pig insulin or coipu insulin.
Signal pattern.
Which one? Signal. They may also, yeah, they may also have actually, but yeah, you can see the answer is here. So that's why I was saying that you have not taken a look at the slides. The answer is that guinea pig and coipu insulin do not bind to two zinc ions. Okay, while other insulin molecules from other species, they do have to. So there is less functional constraint on insulin from guinea pig and coipu, and so they evolve faster than those from other species. So you can take a look at the evolution of the insulins of human, mouse, and guinea pig, and the changes are indicated by these vertical arrows. So wherever changes are observed in guinea pig relative to human and mouse, so there are several positions where substitutions have happened. Okay, and again, this is signal peptide. So same thing, signal peptide, C peptide, B chain, A chain. Okay. Okay. So, and one of the most influential hypotheses in the field of evolutionary biology was molecular clock hypothesis that was proposed in 1960s, sometime 1962 or something like that. So around that time, by Linus Pauling, Emmanuel Margolius, and others. So Pauling, I mentioned that Frederick Sanger, he got 2 Nobel Prizes, both in chemistry. I mentioned during the first lecture. And Linus Pauling also, he was professor of chemistry at Caltech, California Institute of Technology, and he also got 2 Nobel Prizes, one for chemistry and the other, for?
atomic theory.
No, yeah, for peace. Yeah, not science, for peace. So yeah, he got 2 Nobel Prizes, one for chemistry and the other for peace. So very popular person, chemist. So they proposed that for every given protein, the rate of molecular evolution is approximately constant in all evolutionary lineages. So consider proteins from a family like insulin family. So insulin evolves with a constant rate across different lineages. Okay, so they observed when they were analyzing the sequences the protein sequences. So by that time, there were dozens or hundreds of sequences, and they consider a small abundant proteins such as globins, cytochrome C, and fibrinopeptides. So they consider these from different lineages, and then when they analyzed, they found that cytochrome C evolves at a constant rate. You can see the slope is right. So all these I will discuss further. All these points lie along the line, and you can get the slope. And so it evolves slower than hemoglobin, and then fibrinopeptides evolve. Hemoglobin evolves faster than cytochrome C, and fibrinopeptides evolve faster than others. Okay, than hemoglobin and cytochrome C. So if you see. Yeah, I would like you to appreciate what this figure is conveying. So here is the time since plants and animals evolved from their common ancestor. So plants and animals evolved from their common ancestor 1,200 million years ago. Okay, and so on the y-axis is percent amino acid changes. Okay, so if you consider the sequences of plants and animals, you will see. Okay, let's look at this on the y-axis. So animals and plants. So 60%. So if you compare sequences of plants and animals, you will see 60% substitution, 60% changes. Okay. So these organisms shared last common ancestor 1,200 millions of years ago. So they are highly diverse. When you compare these sequences, you will see only 40% identity, 60% changes, mutations, okay, or substitutions. Okay, great. So, and this is giving you a sense of the rate of evolution. So, let's see next. So, this is for vertebrates and insects. So, vertebrates and insects shared their last common ancestor 600 million years ago. And if you look at their sequences, cytochrome C sequences. So I'm just focused on this cytochrome C. So if you compare their cytochrome C sequences, so number of mutations or percent mutations is 30%. Okay. So from 1200 million years to 600 million years, and percent mutations also got halved, right? So 1200 million years halved, so 600 million years, and then the number of mutations, or here percent of number of mutations per 100 aligned residues, so percent mutations, that also changed from 60 to 30, right? So as you further look in other lineages, it is changing at this proportion. Okay, so which indicates that this protein cytochrome C is evolving with a constant rate in different lineages. That's why these points are on the straight line. Okay, so 1200 million years, and how many changes have happened in 1200 million years? 60% changes. Okay, so how many years it will take for 1%? So 60% will happen in 1200 million years. For 1%, how many years it will take? 20, right? So 60% in 1200 million years. Okay, so 1% would be 1200 divided by 60 is equal to 20. Okay, so 20 million year. So similarly, for hemoglobin, it takes so for 1% change, it takes 5.8 million year. for fibrinopeptides, it takes 1.1 million years. Okay, so fibrinopeptides are evolving faster than hemoglobin and cytochrome C. And this is again, you will say that, okay, there are less functional constraints on fibrinopeptides compared to hemoglobin and cytochrome C, and that's why this is happening. So if it is so, so here is the rate actually, and of course, for rate you would want to compute the number of substitutions per site per year, something. Okay, you can compute that. You have the data, you can compute. So, and this is very interesting that if proteins evolve at constant rates, then they can be used to estimate the times that sequences diverged, which means that you can time the evolution. You can know when two organisms evolved from their common ancestor, and that's great. And that can happen from this hypothesis, that can happen if proteins evolve at constant rate. So then you can do that. You can time the evolution. You can infer when two organisms shared a last common ancestor, Okay. So, and this is analogous to dating geological specimens by radioactive decay. Okay. So, I would like you to try timing the evolution of plants and animals. Okay, I have given you that 1% changes happen in 20 million years. Okay, and you have to let me know when plants and animals shared their common ancestor. So what will you do? So you are given this. So this is the information given to you. So rate of evolution, it is constant given to you, and this is for cytochrome C. And then I am asking you, so cytochrome C evolving in plants and animals. So 1% change is happening in 20 million years. And I am going to ask you to now time the evolution of plants and animals. Tell me when they shared their common ancestor. So what will you do? No, I don't know. That is the question. That is what I want to know. So that is unknown. Okay, that is unknown. Now tell me the what you would like to do. So you are given this, you are given this, and you don't know when they shared a last common. That is an answer, and I want to get to that answer. Yes, go ahead.
Will check the similarity between plants and animals and how much difference between the.
Okay, very good. So you would want to compare the cytochrome. So this is for cytochrome C. You would want to compare the cytochrome C sequences from an animal and a plant. Okay, align them, which you have been doing. Align them. Align cytochrome C sequences from a plant and an animal. Align them. Okay, and then BLAST will do for you, or any alignment algorithm. Even you can use global alignment algorithm. You can use. And then you will come to know the percent divergence or percent changes. Okay, and when you will do that, you will come to know that there is percent change. Okay, and we know actually that it's 60. So 1% change takes 20 million years. How many years will 60% change take? So 1% 20, for 60% 20 multiplied by 60. So then you will say that plants and animals shared their common ancestor 1200 million years ago, right? So yes.
Let's suppose you don't know if plant and animals share common ancestry between them is 1200 million years ago.
Right.
Then how will you know if 1% difference similarity 20 million?
So how we know that?
And how is that?
Yeah. So people have been timing evolution of organisms using different methods, not just this. And you know, actually, from using geological, also using, I think, geological specimens. So, if you are able to gather the information about the rate of evolution of the sequences, then you should be able to, because it is difficult to get this, okay. But once you have gathered this for any organisms, if you have got this information, then it is easy to infer, because now these days it is easy to get biomolecules and sequence them, okay. And there are so many different lineages, because this is constant, so you should be able to time for any lineages. But of course, in first place you have to get this through some other methods, and that will be complicated, and it is just not possible to do this for n number of lineages, because that is something for which you need some data, right. Whatever, geological specimen or whatever, there are several methods for timing the evolution of organism, not just the sequences, but those are difficult to do. But if you come to know that the rate of evolution is constant, then it has made your life easier. You have to just get the sequences, and if that is known, then you can just time the evolution of the organisms of certain lineage, okay. Okay, so next. Positive selection and negative selection. Positive selection occurs when sequences undergo significantly higher rate of substitution, which means that there is a need of a new trait to the organism. So, rapid evolution is selected in the because it may confer new traits. So, those changes will result in creation of new proteins, and those proteins can make that organism fitter in a certain environment, okay. So, and that change will be selected for, because that change is making the organisms fitter in a certain environment, okay. On the other hand, there are changes that can result in disruption of proteins, organisms may die. Those changes will be selected against in the course of evolution. So, that is negative selection, okay, and that happens slowly. Okay, otherwise selection is neutral. So, and now we will get to, with some discussion on evolution, we will get to phylogenetic trees. And first, the nomenclature of trees. So, trees have topology and branch lengths. So, what does topology mean? Topology, branching patterns, okay, just branching patterns, and of course, then branch lengths. So, you have branching patterns, you have branch. What are the lengths of the branches? So, we will now describe the parts of a tree. So, here are the trees depicting evolutionary relationships among organisms. Trees could be based on nucleic acid data or protein data, okay. Okay, so let's get next. So, trees could have taxa actually. So, these are the leaf nodes, the descendants actually from ancestors. So, they are represented by leaf nodes or terminal nodes, and at the terminal nodes you have A, B, C, D, E, and these are the sequences that you would want to begin with to understand their relationships, okay. And then the ancestral nodes are inferred. So, ancestors are inferred, and as you construct the tree, you go to the last common ancestor, which is the root, okay. Okay. So, and taxon could be groups. So, taxa actually. Taxa could be groups. So it could be species or genus, a taxon, or family, or it could be some operational taxonomic unit. Have you heard about OTU? So what is an OTU? So I'm sorry.
16S RNA, 18S RNA,
One,
IPS.
Six.
one group from one.
Group of organisms. Okay, so yeah, group of organisms that are having say X% or more similarity with each other. Okay, for example, you said 16S. Okay, so when you are considering prokaryotes or bacteria, you consider 16S as you said, right? So you would want to compare them, and if the identity is 97% or more, then they are grouped together. So all pairs within a group will have identity 97% or more, and that is called operational taxonomic unit, right? So this is again with some heuristic they have this number 97 or 97%. So that's an operational taxonomic unit, and it was done because I think it is very hard to classify prokaryotes, bacteria and archaea. They keep revising it because the species boundary is hard to define in bacteria or archaea, unlike eukaryotes. Okay, so one way was to just consider 16S and group them. So it is possible that that group may have all those species that belong to a genus, or maybe strains that are arising from a species. But I think with this 90% threshold, it does include apparently more divergent by taxonomic classification, more divergent organisms. So maybe different species can get grouped when you have this 97% threshold. So which is okay, and this is done for now these days, metagenomic sequencing and then classification of the organisms based on the metagenomic sequences. Okay, so you understand these taxa, and then the nodes. So leaf nodes are the terminal nodes. So they could be the sequences representing the sequences that you want to analyze, or they could be the organisms that you are interested in analyzing and understanding their relationships. So nodes are what are nodes? So nodes contain taxa, right? Taxa, and edges indicate the distance between the taxa, right? So edges connect the nodes, indicating the evolution of a descendant from an ancestor, right? Okay. Okay. So the branches can be unscaled. Unscaled means that the length of these edges from this ancestral node, they are same. Okay, so which means that A and B have evolved with a constant rate, same rate from F. Okay. On the other hand, branches can be scaled, which would indicate that A is evolving faster than B, okay, because this length is indicative of number of changes that have happened since the evolution from their common ancestor. Okay, that is, so if more changes have happened, then the age would be longer. Okay, so branch lengths are proportional to number of amino acid changes. So bifurcating node. So bifurcating node means that an ancestral node will result in how many descendants? Bifurcating, 2 descendants, okay? While multifurcating would indicate that an ancestor will give rise to multiple, more than two descendants, okay? Yeah, in evolutionary biology, multiple means not more than one, more than two, okay, because of pairwise and this concept. Okay, that's fine. Okay, clade. So definitions of the features of a tree, phylogenetic tree. So what is a clade? Clade is a group of organisms that have descended from a common ancestor, include common ancestor also. Okay. So if you include common ancestor F, then their descendants are A and B. Okay. And the clade is ABF. And now if your common ancestor is H, so the clade would be CD. Okay. If the ancestor would be G, then you have to include all descendants from G. So that would be ABF, CDH, include G also. So this is also a clade, a larger clade. Okay. And then you'll include everything. If you include I, then you have included everything. Okay. So the biggest clade that you can have. Yes. Yes, this is monophyletic. So tree roots, and what is a root? Root is the last common ancestor. Okay, so you can see actually the root. So some trees are rooted. The last common ancestor here is, look at this tree. The last common ancestor is 9. So 9, and this is a rooted tree. A tree can be unrooted. For example, this is an example of an unrooted tree. We don't know where the root is. And often when you make such a tree, you would want to use an outgroup in order to root a tree. Okay, so synonymous and non-synonymous changes in the human, mouse and horse beta globin coding region. So what are synonymous changes? What are synonymous mutations? Yeah, proteins, yeah. So nucleotides can change, but amino acids will not. Okay, that is synonymous substitution. And non-synonymous substitution, yeah, nucleotide changes, and because of that amino acid changes. Okay. So that's non-synonymous, which indicates that protein sequence will change because amino acid changes happen. So let's look at the changes happening in the human, mouse and horse beta globin coding region, and let's focus in the coding region, coding sequence CDS. So that will begin with ATG. So always remember, if it is CDS, it will begin with the start codon. In eukaryotes, almost always ATG, and will end with one of the 3 stop codons, TA, TAG, TGA. Okay. So let's look at changes in the coding sequence. So is this change synonymous or non-synonymous? So you said synonymous? Non-synonymous, right? Because there is a change or mutation in human beta globin coding region, and which is the AG substitution. So you can see that in mouse and horse it is G, in human it is A, and that results in a different amino acid K in human. Okay. So the next one, this one synonymous, non-synonymous, non-synonymous. Next one synonymous, non-synonymous, and the last one synonymous. So you understand. And you can see that, of course, because of more functional significance of the coding sequences, So more functional constraints, there are fewer changes. On the other hand, see the regulatory regions, so the promoter sequences upstream of the start of the genes, you can see that there are many changes because these regions have less constraints. Okay, of course they are important, they have functional significance. So there are conserved sites there as well that you can identify using a method called multiple sequence alignment. So I think these types of questions should be there in the test. Okay. Tell Dr. Shukla that include these questions. Okay. So okay, very good. So if the synonymous substitution rate is greater than the non-synonymous substitution rate, then there would be negative selection or positive selection. If synonymous substitution rate is greater than non-synonymous substitution rate, negative selection or positive selection? Negative selection, right? Because the pressure is not to change the protein. So negative selection. If a non-synonymous substitution rate is greater than synonymous substitution rate, negative selection or positive selection? Positive selection, because protein is changing, right? Non-synonymous rate is higher than synonymous rate. So protein is changing, and that is helping organisms because it may be conferring novel trait due to organisms in a dire condition, okay, difficult environment. So then it is important for organisms to acquire novel trait to survive, otherwise all organisms will die. So those changes that can confer enhanced capability for an organism to survive and thrive, they will be selected for, okay, and that will come when proteins will change. Okay, very good. The fundamental basis of a phylogenetic tree is a multiple sequence alignment, and we did that. We learnt about multiple sequence alignment, different methods that are used for aligning multiple sequences. Consider the alignment of 13 homologous globin proteins, and they are myoglobin, what are these? Multiple alignment of myoglobins, alpha globins, beta globins, and they are, I think, coming from different organisms. I can see dog, KAN is what? Kangaroo, or I don't know actually, soybean and so on, okay, from many different organisms. So and you can see gaps, there are terminal gaps, and there are many substitutions. But multiple alignment algorithm can capture some characteristics that can help in classifying different proteins, for example, okay. So let's look at those positions. So those positions that can help discriminate between myoglobin, alpha globin, beta globin, and just globin, soybean globin or insect globin. Okay, just forget this. Just consider these myoglobin, alpha globin, and beta globin. So look at these positions indicated by open circle. Okay, so you can see that in myoglobin the residues are only G. Okay, look at this column actually indicated by this open circle. So in myoglobin the residues are G, in alpha globin it is A, in beta globin it is E. Okay, so these are informative sites, and those sites got captured by multiple alignment, and these sites are discriminating between these three types of globins. There are sites that are highly conserved, but that will not help in discrimination, because everything is same actually in that column. So how you can use that for discrimination? So they are not informative so far as classification is concerned, right? So it is important that multiple alignment should capture those informative sites that can help in classification. So okay, so this site and this site also. So if you look at this site, you have the residues I for myoglobin, E for alpha globin. So EE, where is it? EED, okay, that's okay. And then G for beta globin. Okay, so almost I think a good column that can help in discriminating between these three proteins. So if multiple alignment and a good multiple alignment method should able to provide you such an alignment where informative sites are apparent. Okay, you can take a look and you should be able to say that okay, this is a good an alignment, a good alignment. Okay, so after you have performed alignment, which we did yesterday, the next stage is to compute the divergence between the sequences in the alignment, in the multiple alignment, how diverged sequences are. Okay, so you have to estimate the degree of divergence. So one approach could be that okay, I will look pairwise in the multiple alignment, so two sequences, and I will compute the divergence of the sequences, so all pairs in the multiple alignment. And one very simple approach is to get the mismatches in the alignment of two sequences. Okay, so get percent mismatches or percent changes, that is called Hamming distance. Okay, where n is the number of changes in the aligned regions, and aligned regions have a total n residues. So out of n there are out of capital N there are n changes, and n is a small n. Multiply by 100, that is the percent change. Okay, that is the Hamming distance. Okay, so and something that we had been discussing this, that when you perform alignment, of course you observe changes, but observed changes do not equal genetic distance, because there could be many changes that have happened, but that cannot be observed when you perform alignment and when you look at positions that have mismatches. We discussed about this, we discussed about back mutation, and also some positions could have undergone many mutations, many rounds of substitutions which are unknown. So models have been developed. This field, evolutionary biology, is a lot mathematical. If any of you are interested in this, it's a great field, mathematical, but at the same time very fascinating. So many mathematicians got drawn into this, and the methods that we are discussing today, some of those were developed by mathematicians from around the world actually, and they collaborate and they fight with each other. It's really because this is based on hypothesis, you don't know what happened. So there could be more arguments for and against certain positions or hypotheses. So sometimes a lot of heated arguments happen, and I have attended many conferences where people have become very angry with each other because they have their view and the other group is saying that okay, you are not right, and sometimes they say bluntly. They fight kind of with each other. So tree war. Okay, so there was a slide on tree war. So and I don't consider myself evolutionary biologist. So anyway, I enjoy the discussion and the fight that those people have. Okay, so so genetic distance involve mutations that are not observed directly. Okay, and there are models that have been developed for including the unobserved mutations. Okay, and one of such models is Jukes-Cantor model, and here is the divergence between two sequences proposed by Jukes and Cantor, where p is the Hamming distance. Okay, this is the formula, and one of the limitations of this formula is that it assumes that the rate of transition is equal to rate of transversion. Transition, you know what is transition? Purine to purine and pyrimidine to pyrimidine. Okay, and transversion is cross. Okay, so this assumes that okay, so this is transition between purines and transition between pyrimidine nucleotides. Okay, a cross transversion. So, this model assumes that the rate of transition is equal to rate of transversion, alpha is equal to beta. Okay? So, it is all alpha, rate is same. Later on, Kimura proposed two parameter model, where he proposed that alpha is not equal to beta, and he derived a very complicated formula. I have not brought that for you, do not worry. So, it is there. So, and people use these divergence formulas to compute the divergence between sequences from multiple alignment. Okay? You would want to compute this to make a tree, phylogenetic tree. Okay, so here are the 5 tree building methods: UPGMA, never joining, maximum parsimony, maximum likelihood, Bayesian. Okay, UPGMA and never joining are distance based, maximum parsimony is character based, maximum likelihood and Bayesian are model based. Okay. So, distance based methods are based on some distance metric, where you estimate the relationships between organisms in terms of distances between them, and distances again could be molecular. Okay. So, two proteins from those organisms and how distant they are. So, you compute similarity score, and now you also know that the similarity score can be converted to distance score when you are making a tree. Okay. Okay. And character based methods are different in the concept that the whole idea of character based methods is that let us make a tree that can have minimal number of substitutions or mutations, that can explain the divergences in those sequences that you have used to make the tree, because the whole idea is that nature is parsimonious. So, there should be minimal number of mutations that can result in different organisms. Okay. So, relationships between organisms can be interpreted by constructing a tree with minimal number of substitutions. Okay. Okay. Continue. Okay. Okay. So, so here is the UPGMA approach, and this is very simple approach, and this is called unweighted pair group method using arithmetic mean. Okay. So, this has been frequently used for making a tree topology, and I would not ask you to make a tree using UPGMA method because of some limitations of this, but you would want to understand what this method is, because this is a very good approach for understanding how a phylogenetic tree can be made. So, given 5 objects, and those 5 objects can be 5 sequences, you identify 2 sequences that are most similar to each other among all these, and let us say that those 2 sequences are 1, 2, okay, that are closest to each other among all. So, you would want to consider all pairs and then identify the pairs that has the closest sequences. Okay. So, that pair is 1, 2. So, you will place 1 next to 2, and so you are making tree, and you will create an ancestral node 6. Okay. Okay. Next, now you have, so you started with how many sequences? 5. So, and say that okay, you have 5 groups each containing 1 sequence. Now, you are left with how many groups? 4, where one group contains 2 sequences and the other groups contain 1 sequence each. Okay. Now, you repeat this, repeat the comparison. So, again pairwise comparison, where you have 4 groups, one containing 2 sequences and the other groups containing 1 sequence each. Perform again pairwise comparison and identify the groups that have the closest sequences. Okay. And the next group could be 4 and 5. So, you will put 4 next to 5 and create the ancestral node 7. Okay? And you are also computing the distance. So, the distance between 1 and 2, which is D12, D12. So, this is 1 and this is 2 and okay, 6. So, this distance is half of D12, okay, and this is, of course, this is also half. So, the vertical line. So, that if you add them up, so they should add to D12. Okay. So, from the ancestral node, so which is 6, so half D12 plus half D12, D12. Okay. So, that is the distance between the two sequences 1 and 2, and similarly for this. And now you repeat this. Now you have 3 groups, 2 groups with 2 sequences and 1 with 1 sequence. Okay. So, let us see how they will get grouped. So, this is just an example. So, when you will repeat this, you can see that 3 and a group of 4 and 5, they are most similar to each other, closest to each other. So, they should be grouped. So, now 3 has grouped with 4 and 5. Okay. 4 and 5 represented by ancestor 7, and when you have grouped 3 with them, now you have created common ancestor 8. Okay. So, keep repeating this. So, now when you will repeat, then so iterate this. When you will iterate, then now these 2 groups will come together, right. So, now everything in one group. So, which is the last common ancestor number 9. Okay. So, thus you have constructed a rooted tree where root is 9. Okay. And this is a simple method for constructing phylogenetic tree. So, it is always rooted. It assumes that the molecular clock is constant. So, proteins from an ancestral node evolve with constant rate of evolution. Okay. So, proteins are evolving with the same rate from ancestral nodes. So, while this is simple, it is less accurate because of the reason that I will tell you next. So, let us consider this tree, and in this tree you can see that from the ancestral node 1 has evolved rapidly, but 2 is evolving slowly. Okay? And on the other side, 3 is evolving very slowly and 4 has evolved rapidly. When you will use UPGMA method, because these 2 have not evolved much, they will group together. Okay? They will be placed next to each other using UPGMA because they are more similar than others, because they have not evolved much when they started evolving from the common ancestor. Okay? So, they will get placed next to each other, and then 4 and then 1. So, UPGMA will generate this tree, while we know that the evolution happened this way. 1 is closest to 2, 3 is closest to 4, and then they have the last common ancestor, which is the root of this correct tree. Okay? Using UPGMA, you will land with this tree, which is not correct. So, people have started developing other methods actually that can account for these long branch lengths, and long branch lengths indicate more mutations, more substitutions. So, rapid rate of evolution since diverging from their common ancestor. Okay. Okay. So, and here is one definition actually. So, additivity of distance. So, given a tree, a distance measure is additive if you want to compute the distance between 2 nodes 1 and 4, and that can be obtained by adding the lengths of the branches that connect those 2 nodes. Okay? So, if you want to compute the distance between 1 and 4, that can be computed by summing the lengths of the edges that connect 1 to 4. So, those edges are this one and this one. Okay. So, if you add them up and that is the distance between 1 and 4, then the distance metric is additive. Okay. Okay. So, if distance metric is additive, so here it is. So, if distance metric is additive, then a method called neighbor joining that you may have heard about can produce the correct tree even if you have those long branches. Okay? Even if you have differential rates of evolution from the common ancestor. So, even then this method will produce the right tree. So, will produce this tree given the sequences 1, 2, 3, 4. Okay? So, the whole idea was that, and there is a proof in Darwin et al. Book. If you are interested, it's mathematical. So, it is in the appendix. You can go and see the proof if you want further information regarding this. So, what they did actually, they recomputed the distance between two nodes, okay, ij. So, and dij is the same as in UPGMA, okay, but they compensated with the term Ri or Rj. So, Ri, is it I? Okay, yes. Ri is the average of the distances from a node. So, for example, you want to compute the distance between 1 and 2 using neighbor joining method. Okay. So, first you will compute D12 as in UPGMA, and then you will compute R1. So, R1 will be obtained by taking the average of the distances from the other nodes. So, 1 compared with 3. So, distance between 1 and 3 and distance between 1 and 4, take the average of this, that is R1. Okay. R2 would be the distance from 2 to 3 plus distance from 2 to 4 divided by 2. So, the average of these two, that would be R2. Okay. So, when you subtract these distances, that will compensate for the long branch lengths. There is a proof, you can, if you are interested, you can do it. Okay. Go through, that is very interesting. So, this method because of this reason has become really very popular method in constructing tree, and that is why I said that if you have to construct a tree, do not use UPGMA because of this problem. You would want to use neighbor joining because it will give you the right tree, even if the distances are not additive, may still produce a good tree, not even if it is not a right tree or correct tree, it will be a good tree. Okay. If distances are additive, it is guaranteed to produce the correct tree. Okay. Okay. So, next, character based method. So, and as I told you, let us see with the example. Okay. So, there are 4 sequences and you want to create tree topologies. Okay. So, you want to create different tree topologies and identify the topology that has the minimal number of substitutions or mutations. Okay. So, these sequences, they are at the leaf nodes. So, AAG, AAA, GGA, AGA. Okay. And here are 3 topologies shown for them. So, AAG next to AAA, and the inferred common ancestor is AAA, and GGA placed next to AGA, inferred common ancestor is AGA, and the last common ancestor that was inferred is AAA. Okay. And let us look at the number of substitutions in this tree. So, from AAA to AAA, no substitution, right? From AAA to AGA, how many substitutions? 1. Okay. And then from AAA to AAG, how many substitutions? 1. And then AAA, no substitution. From AGA to GGA, how many substitutions? 1. From AGA to AGA, no substitution. So total number of substitutions 3. So that is the cost. Okay. So let's consider this topology, and you have the inferred sequences, all are AAA, and from AAA to GGA, how many changes? 2, no change, 1 change, 1 change. So total change 4. Okay. So similarly here, total change 4. So which is most parsimonious? Which among 3 is most parsimonious? First one. Yeah, because only 3 mutations can explain this relationship, and this is just based on the parsimony theory, and parsimony theory is really very popular not just for the purpose of constructing tree, but in other domains also. It's really very interesting. So get the most parsimonious solution. Okay. Okay. So Bayesian method. Do you remember Bayesian theorem? We did it yesterday. So what is this? What is Bayesian theorem? Okay, it's fine. I will write at the top. Okay, so I can come. Yeah, it's okay. So yeah, just one formula. So probability of x given y is equal to probability of y given x multiplied by and sum over x. Same thing here. So x here is tree, okay, and y is data. Okay, and now you know you can fill this. So basically this method is computing the posterior probability of the tree given the data. Data is evidence. After having seen the evidence, you can include the prior information here. This is the prior probability. Okay, but the prior probability may change when data are given. Data here are the evidence. Okay, so one can compute the posterior probabilities of the trees. So say that there are n possible topologies. You can compute the posterior probabilities of posterior probability of each of these topologies, and when you add them up, they should add to, when you add posterior probabilities, they should add to 1, right? We discussed 1. Okay. So once you have computed, and there are methods for computing posterior probabilities, you might have heard about MCMC something. Have you heard? No? Okay. Markov chain Monte Carlo. Okay. Okay. So there are methods actually. You might not have done it, and I don't remember when I did it. So, but if you happen to, you will. Okay. So it will help you compute the posterior probabilities, and then which one you will select, which tree you will select if there are n trees. If you have n tree topologies or n trees, so topology and branch length, so n possible trees, and you have computed the posterior probability of each of these trees given the data, which one you are going to select? You computed for all, then which one you would select? Okay, in the gene prediction problem, which one we were selecting? So we computed the posterior probabilities for coding in the 3 frame, non-coding, and then gene shadow. Okay, and when we had to predict gene, then which one we were selecting? Which one? Highest probability. So among these 7, one that has the highest probability value, right? And same thing you will do here. Same thing. The tree that has the highest value of posterior probability, you are going to select that tree. Okay? Because that tree is most likely the right tree that explains the given data relationships in given data. Okay? Okay, so this is the concept of Bayesian phylogenetics. Okay, clear? Okay, so the next approach, and here is a tree made using Bayesian method, and this is a famous method for constructing Bayesian tree, phylogenetic tree based on Bayesian method, Mr. Bayes. Okay, developed by researchers at University of California, Berkeley. Okay, so here is a tree made using Mr. Bayes, which was published in Science, and this is a tree of animals, different animals. Okay, so yeah, it has been a while since this was published, but I think it might have been very interesting. That's why I got published in Science. Okay, so yeah, Bayesian method is used frequently in constructing trees, and one of the most used is also maximum likelihood. Okay, you might have heard about ML method or maximum likelihood method. So this is basically this thing. Okay, so let's write here this side. So likelihood, so probability of data given tree. Okay, just compute this. So basically, which phylogenetic tree? So phylogenetic tree means both tree topology and branch length, which has the highest likelihood of producing this data. Okay. So you will compute this probability of data given the tree. You remember you computed probability of sequence given model, right? Markov model, you computed that. This is the same thing. You compute given a tree. So given a tree means tree topology and branch length. Compute the probability of the data given that tree. Okay, I am not going to go into detail of that, but that will give you the maximum likelihood tree. So
Can we break for?
yeah, just I am last slide almost, so I will break it, and then so the methods that use actually, or I should say the programs that use this method are tree puzzle, Mega5, POP, and Phylip. Okay, so last stage 5. So once you have constructed a tree, how you can be confident that your tree is good? Okay, a statistical confidence over the tree that you constructed, and the method is called bootstrapping. Okay, and it begins with the multiple alignment. So you use the multiple alignment to construct a tree. Okay, now what you do, what is bootstrapping? So bootstrapping is based on random sampling. So you will sample from the multiple alignment a column randomly. Okay, so say that you have a multiple alignment with 100 columns. So you will sample a column and say that you have sampled column number 49. So in the bootstrapped sampling, this 49 becomes column number 1. Then replace it, put it back, then sample again. So now you may sample column number 30 from the original. This will become column number 2 in the bootstrapped multiple alignment that you are making. Okay, so do this as many times as there are columns in the original multiple alignment. So I said there are 100 columns, so you will have to do this 100 times. So thus you will make a bootstrapped alignment, multiple alignment with the same dimension as the original one, right? So now make a tree using that multiple alignment. So that would be bootstrap tree 1. Make 100 such bootstrap trees. Okay, and then ask. So let's go to the tree, and then ask. Let us say that you first made this tree. Okay, after you made this tree, you want to assess how good this tree is. After this, you did bootstrapping, and using bootstrapping, you made 100 bootstrap trees. Okay, and then you will ask that of 100 bootstrap trees, how many have 11 placed to 9, which means that this clade also represented in them. Out of 100 bootstrap trees that you made, how many have this clade represented in them? And this clade has 9 placed next to 11. Okay? And when you will check, you will say that everyone has. Okay? 100 out of 100. So the bootstrap confidence would be 100%. Next one. So when you look at this clade where 9 is placed next to 6, out of 100 bootstrap trees, how many have this clade represented in them? Okay, 98. If 98, so 98%. Okay, so you can do for all these clades, bigger clades, smaller clades, and so on, and you place bootstrap confidence values over all these clades, all these nodes representing ancestral nodes representing the clades. Okay. So typically it should be more than 70%. Then you have confidence that the clades are statistical. Yeah, how many of you have done analysis of RNA-seq data? None? Oh, okay. So what kind of data are RNA-seq data? Transcriptomic data. And transcriptomic data, you understand, I think we discussed it. It is one of the omics data. DNA makes RNA and you get RNA transcripts. That data is transcriptomic data. Okay. So RNA sequencing or RNA-seq technology is relatively recent technology. Before that, microarray was used, microarray technology, but it was soon replaced by maybe 10-15 years or so. So when I say soon, actually, so I'm thinking about that time scale when technologies are replaced. So it got replaced by RNA-seq because this technology is really very powerful and very useful. With RNA-seq technology, you can perform unbiased analysis of transcriptomic data, which means that you are not biased by, for example, what are known. So in microarray, you have the probes for the known genes, right? So you are constrained by the knowns. In RNA-seq, actually, you consider all transcripts that are arising from some experiment. That could be some condition under which an organism has been subjected. So you will collect all RNA transcript data, and then you would want to quantify the abundance of RNA transcripts. And some of the transcripts could be arising from genes that are not known. So you may identify some new genes. So gene discovery that we discussed. Okay. So gene discovery through RNA-seq, or you can find novel isoforms. Isoform, you understand? Isoform. What are isoforms? I'm sorry? Similar? No, not similar actually. Isoform. So,
Variations or versions.
oh, variants. Oh no. So here actually, I am uh, uh, thinking about alternative splicing. So alternative splicing, you understand? What is alternative splicing?
Intern getting removed and extern getting ligate.
Yeah.
Sometimes one extern may stay one
Yeah.
and three, sometimes two and three.
Right, right. So, which is actually a gene may give rise to multiple proteins through a process called alternative splicing. So, where an exon, for example, sometimes it can act as coding, sometimes it can act as non-coding. So, when it is acting as non-coding, it will get spliced out. Okay. So, and because of this, isoforms can be generated. Okay, splice isoforms. So, RNA-seq can help in identifying the isoforms. So you can get the data, and there are computational tools that can resolve the isoforms that are there in the data. Okay, so let's keep going with these slides. They are not many, so I want to just appreciate what this technology is. Okay, so this is the workflow, and we will go one by one. So there might be a biological question. For example, I got a disease, and I just want to know which gene is responsible for my disease. Okay, so that is a biological question. And then I will collaborate with a wet lab biologist. I will say that let's design an experiment. Okay, so next step, experimental design. So I will say let's design an experiment. Collect RNA from me, but collect in triplets. Uh, so you understand replicates? Okay. So we want to understand biological variability. So that's why we want to get data in multiples of samples actually, representing different times could be, or different some different conditions or whatever you say from me. So at different times, or maybe the same region, but get it in triplets. So three times you get 3 samples, and then 3 samples from healthy individuals. Okay, so that is experimental design. So you are thinking about how many samples you want to get. So maybe 6 samples actually. So that's experimental design. And typically, if it is very expensive, then 3, but can be more. Can as many as possible, maybe 6 samples or even 10 samples if it's cheaper, because that gives more statistical power. Okay. Okay, and once the experiment is designed, then isolate RNA. Okay, so RNA will be isolated from me as well as a healthy individual or healthy individuals, multiple healthy individuals. Okay, so then here are the steps for RNA-seq. Once RNAs are extracted, so RNA library is made. So I will show you this again in a moment. And then the RNA library is subjected to sequencing, next generation sequencing. So this can sequence multiple of the RNA reads, and these reads are short fragments from RNA transcripts. Okay, so it can sequence millions to billions of these short sequences arising from RNA transcripts. Okay, so once these sequences are obtained, they are mapped back to the genome, and then mapped back to the genome, and then quantify the abundance of reads for each of the genes in the genome, right? So when they are mapped onto the genome, you know that how many reads map to this gene, how many map to this one, and so on. Okay, so you will do this for healthy first, and then you will do this for me, actually, a sick individual, some disease. Okay, and then compare. So you want to identify which genes have difference in the abundance of reads for healthy and me. Okay, between healthy and me. So which have the differences. So for example, a gene may have RNA abundance of say 100 in healthy, and in me 1000. Okay, so how many fold more in me? 10-fold more in me. So it means that that particular gene is producing more RNA transcripts in me. Okay, and the disease that I am having may be arising from that. So again, that's a hypothesis, because that is indicative of something not right, because in healthy it is 100, in me it is 1000. Okay, So this is what RNA-seq helps in inferring, basically quantifying the abundance of RNA, so RNA transcripts in terms of the reads that are generated from this technology. Okay. So reads are mapped onto the genome, and then the expression level is quantified. So more is the abundance of RNA transcript, more is the gene expression. Less abundance, less expression. So expression, when a gene expresses, it makes RNA transcripts. So more expressing, it will produce more RNA transcripts. Okay, very good. So this will result in hypothesis testing. So you have found that there is a difference in RNA abundance in a certain gene between healthy individual and me. But you would want to assess whether this difference is statistically significant or not. Is it arising because of a biological reason or just by random biological variability? Okay, so you want to assess the statistical significance of this difference. So that's hypothesis testing. Okay, and you may also want to perform exploratory statistics. So perform clustering analysis. For example, principal component analysis of the samples. So 3 samples from me and 3 from healthy individuals. Okay, so perform PCA. You would expect that my samples, they will group together and will show as a group in the PCA plot. And those from healthy individuals, they will group together far from me. Okay, so you'd expect that. And also, once you have obtained the genes that are expressing, or just the abundance of RNA transcripts in terms of the reads, counts of reads that mapped onto those genes, you can perform clustering of the genes in terms of their gene expression patterns. So basically, in terms of the abundance of the RNA transcripts arising from them. Okay, so if 2 genes are expressing similarly, they would have similar pattern of transcript abundance, right? So they should group together. Okay, and then classification. So you can use machine learning algorithms, and you learned about machine learning yesterday, right? So you have the data coming from, say, different cancer patients. Some have breast cancer, liver cancer, lung cancer, and so on, and also healthy individuals, right? So you have got the RNA-seq data from them, and you can train a machine learning model on this data. Okay, so machine learning model will learn these differential patterns, representing differential patterns of transcript abundance representing different cancers. Okay, so if there are 10 cancers, machine learning will learn these different patterns, 10 patterns arising from the 10 cancers, and then give new samples. It will predict which cancer is represented in which sample. Okay, so that is called classification. So it will classify the samples by cancer types. Clear? Okay, so and then biological confirmation. So if you have identified a gene that is important because it has a differential abundance, you would want to do some wet lab. So design experiment or assay to confirm the function of that gene in the context of what you are interested in. Okay, so this will give you novel biological insights, and it can also be used in diagnostics and therapeutics. So diagnostics, I just mentioned that you want to detect cancer. So classification can help, right? So there is a sample, and maybe stage 1 cancer. So machine learning will identify that sample as stage 1 cancer. So great. Maybe biopsy is not able to, but machine learning model is able to. There could be some therapeutics to address this problem, right? So and the next is actually therapeutics. So RNA-seq can, so this is the workflow. So starting with the RNA. So starting with the biological question and then design and then so on. Okay, and you come to the end here. Okay. Okay, so RNA-seq data are provided in FASTQ format. So there are files, and these are very large files. I have actually the workflow that one of my PhD students, he provided to me, so that I give that to you. And I think you have got the PowerPoint for that. We may not get time to do this, it takes time. But you have the PowerPoint, and I think I refined the PowerPoint, so I can send to you. And if there is a group email, you can forward them. Okay. So I did some refinement because he did not provide the accession numbers of all the samples. So now I have included them. So you can try that later on. And if you are a biology student, once in a while, you may actually have to do such analysis. Because it's an era of big data, and these are the big data, RNA-seq data or DNA sequencing data, these are the big data. So it is possible that you may encounter at some point of time these data and you would want to analyze. So make sure that you have saved the RNA-seq pipeline that we have provided to you, right? So you will try, and if you have difficulty, then let us know. I will ask my student to help with that. Okay. So when I was doing, I was getting some server error, so, and which we have been actually with Galaxy. And he told me that just keep trying. Maybe in the third attempt or fourth attempt, it will work, but it will work for sure. Okay. So his experience. So I was also wondering that why I'm not able to get the answer, why I'm getting the server error. So he said that, yeah, it happens, and it is likely that it happened to him also. And he suggested that we should do it at different times. Maybe in the third attempt or fourth attempt, it will work. Because he has done it, all those samples are okay. He got the results, so you will also be able to. Okay. Okay. So here is the FASTQ format. So each sequence, each sequence is called a read, R-E-A-D, okay, read. These are short sequences, could just be 50 bases or 100 bases, something like that, okay, short sequences. So each of these reads are characterized by 4 lines. The first line is the identifier line, like greater than. You have become familiar with FASTA format. So the first line is greater than, and then the name of the sequence or organism and so on. Okay. That is the first line, identifier line. The second line is the sequence. Okay. And it's a DNA sequence, so it's a sequence of nucleotides. The third line is always plus, maybe for some reason. It is always plus. The fourth line is indicating the base quality score, and it is coded. Okay. So you will have to decode it, and there is information about how to decode. Okay. If you just type FASTQ format, there is an information at different pages on how to decode this. So you can decode, and that will tell you the quality of the base that was sequenced. How good was it? Okay. So then you will have confidence that, okay, this base is good, the quality is good, if the quality is good. Okay. So 4 lines for each read. So typically there are often millions of reads. So you will have 4 multiplied by millions or whatever. That's why these files are really very, very big. So, and here is the formula for quality. So this is called pH rate quality. This is the score. So PE is the probability of incorrect base call. Okay. So if the probability of incorrect base call is 1 in 10, so there would be one mistake in 10. Okay. So then this would be 1 divided by 10, right? And 1 divided by 10 is 10 power minus 1. So log 10 base 10, 10 power minus 1 is minus 1. So minus 1 multiplied by minus 10 is no, minus 1 10. So this is the score, right? 10. Okay. For 1 in 100, the score is 20. 1 in 1000, the score is 30. Okay. So generally here, I think you would say that, okay, it would be good if the error is only 1 in 1000 or 1 in 10,000 or so, right? So now you understand the values of the quality score. Okay, what does 30 mean? What does 40 mean? And this is coming from this formula. Okay, good. Okay, so I think this is a display from a program FastQ that will help you in visualizing the quality of the data. So if they are lying in the green area, so here. So for each position, you have the quality score. So if the quality scores lie in the green area, it means good. If they venture into red area, means bad. Okay, low quality. Okay, so here is the protocol and something that we discussed, that mRNA, and then mRNA is reverse transcribed to cDNA. So cDNA, and then cDNA is fragmented. So sonicate, there are methods for fragmentation, right? What are the methods? Sonication and what else? There could be many methods. Okay. So you may be knowing better. Those who do wet lab, they may be knowing of the techniques or technologies for fragmenting nucleic acids. Okay. So nucleic acids. So here RNA is fragmented. So there could be many fragments of RNA, typically of the same size, and then they are ligated with adapters. Okay. Also sometimes, because these adapters are identifiers, so you can do multiplexing. For example, you can have samples coming from different or individuals, and if you have ligated with adapters, you know which read is coming from which. Okay, so and this is cost efficient. You can do sequencing at the same time of many different samples by this way. Okay, by ligating with the adapter. Okay, and then once you have the RNA library, so this is the library or EST library, whatever you say, then you can sequence them, and each sequence would be 50 nucleotides or 100 nucleotides. So this is determined even before doing this experiment, that okay, what size you want, and it depends on technology also. Some technology can yield long reads. You may have heard about PacBio. Some can yield short reads. Illumina, you might have heard about Illumina. Okay, so once the reads are obtained, they can be mapped back onto the genome. Okay, so here it is shown that this is arising from some gene and it is mapped onto that gene. Okay, so and because this is messenger RNA, so these reads are coming from the coding regions, right? Not from intronic, from exonic. So mature messenger RNA. Okay, so so they are mapped onto the coding region. So you can see that this is intronic region. There is an intron here. So there are no reads that will map onto this. Okay, so these reads are mapping onto the coding sequence, and then you can assess the RNA expression level at each of the position. Okay, at some position there would be many many reads that would map or that would span that position. So which means that there is more expression level at that position, right? So this is what you can. So typically, actually, you can quantify the expression for a gene based on the counts of the reads mapping onto the gene. Okay. So if more reads are mapped onto the gene, then more is the expression level. Okay. So, but when you map, then you have to be careful about some of the things. So it is possible. So let's look at two confounding scenarios. The first scenario is sequencing depth. Okay, sequencing depth. So your sequencing depth could be high. It means that you have sequenced so many times that you have covered the genome maybe 100 times. Okay. On the other hand, for some sample, you have covered only 10 times of that genome. So that's lower sequencing depth, right? So if it is so, then more reads could be arising because of higher sequencing depth, not because of these transcripts expressing more. Okay. So more reads are because of the higher sequencing depth. Okay, more sequencing depth, not because of the gene expressing more. Okay. Okay. So you have to account for this. So basically, you have to normalize with respect to sequencing depth. The second scenario is length bias. So longer transcripts will have more reads mapping onto them. Longer transcripts will have more reads that will be generated from them. So when you will map back, you will have more reads mapping onto them compared to a short transcript, right? And this is because of the difference in the lengths. So that could also confound. Okay. So longer genes can have more reads than shorter genes, right? So this difference is not because of gene expression. This difference is because of the difference in the lengths of these two genes. So you would want to control for that, control for the length bias. So you would want to normalize with respect to gene lengths also. Okay. Okay. So here are the metrics which are the normalization for which has considered the normalization for sequencing depth and length bias. Okay. So the first one is RPKM. RPKM is for each gene or each transcript, okay, reads per kilo base of transcript. Okay. So reads per kilo base of transcript. So you will divide by the length of the transcript and you will multiply by 1000. Okay. That would be reads per kilo base of transcript, and then per millions of reads mapped onto the genome. Okay. So you will divide by all the reads that mapped onto the genome and then multiply by 1 million. Okay. So it would be per millions of reads mapped onto the genome. So this way you have normalized with respect to both length of the transcript and the sequencing depth. Okay. So this is RPKM, and similar to RPKM is FPKM. So FPKM is analogous to RPKM. The only difference is RPKM has often been used for single end sequencing. FPKM is used for paired end sequencing. Okay, and more recently TPM, which is transcripts per million, that has been adopted by the users. TPM. So this is also a normalized metric, and a characteristic of this is that sum of all TPMs in samples is the same. Okay. So yeah, just to understand that you have to normalize, otherwise your comparison will not be fair. You only want to know whether a gene is differentially expressing or not, so you must control for other factors that could be contributing to the difference in the gene expression. So there are two main contributors to the the bias. One is the transcript length and the other is sequencing depth. So you need to control for both, and metrics have been provided, RPKM, FPKM, TPM, that control for these two. Okay. So once you have the normalized counts, you can use the normalized counts for controlling for these. You can use the normalized counts for assessing the difference in gene expression between two conditions. Okay, that is called differential gene expression. Okay. Okay, so I think we discussed about this normalization of counts, and once you compare and you find that there is a difference, as I said that okay, there is a gene in me and I am sick that has normalized count 1000, and a healthy individual has 100. Okay, so there is 10-fold difference. Okay, 1000 by 100. So 10-fold, and this is called fold change. So fold change is equal to 10, and then you would want to assess the statistical significance of this. Is it really significant, or is it just by random chance? Okay, just due to some biological variability you are observing this. So that is statistical significance test that you would want to perform to assess whether the value of fold change is significant or not. Okay. Okay, so and you can see here in this table fold chain, and actually it is log fold chain. Log to help you visualize better the genes that are down regulated and those that are up regulated. Okay, so negative for down regulation, positive for up regulation. Right. So if fold change is 1, then nothing, right? No change. No change. So logarithm of 1 is 0. So that's why. So then the baseline is 0. Below that, down regulated, and above that, up regulated. Okay. So that's negative, down regulated, positive, up regulated. So I will go a little fast because we are running short of time. Okay. So fold change you understand, and significance of fold change. Biologists typically use both fold change and significance value, because sometimes the fold change could be very low, but still that value could be significant, and they are just not interested in those genes. They want to be getting a conservative set of genes that are differentially expressing, so they used more criteria for filtering. Okay, inferential statistics. So this is what is inferential. So hypothesis testing, statistical significance. And here is actually from a paper that we published in 2015. So you can see the reads that were mapped onto different genes. So can you see the reads that are mapped, that are shown in colors? Okay, so you can see that this is GNAT family protein, and in the control there are fewer reads mapping onto this gene. On the other hand, here is the plant actually subjected to highlight stress. Many transcripts were generated compared to control. So you can see so many transcripts whose reads mapped onto the gene. Okay, so is it upregulated or downregulated? Upregulated, right? Because relative to the control, you have many more transcripts that got generated. They are represented by the short reads that mapped onto the gene. Okay. Similarly, WRKI40 got upregulated, KMD1 got upregulated, and this JAT12 also got upregulated. Just to help you understand that, yeah, this experiment was done, and RNA was collected within, I think, 10 seconds or so. So ultra fast changes that happened in the Arabidopsis plant. Yes.
Is we have the tutorial also? No?
Yeah.
So can we take up this one?
Yeah.
In the post lunch?
Yeah.
Because post lunch session will be almost available.
Oh, okay.
Because this is needs some discussion.
Yeah, right,
And,
right,
and
right.
more techniques are there I can see.
Yes,
Yeah, so
right,
that will
right.
be better if you spend more time on this.
On this,
maybe.
right, right, right.
So
right.
I can
Then,
take
yeah.
at 12:30 is there. So
Okay.
we wait for here.
Okay,
And then
okay.
I take the evaluation part.
Okay. Okay, right, right. I think I will finish in five minutes. Okay. Okay, so yeah, let's look at descriptive statistics, which is basically clustering. Clustering is grouping of objects. Okay. So this is something we already discussed, UPGMA. So if there are 5 objects, you can group them. For example, A and B are closest to each other, so they are grouped together as AB into one cluster, and then you will repeat this recursively, just like UPGMA. Okay. Then D and E will get grouped, and then C will group together with D and E, and then all will group together. Okay. So this is called agglomerative clustering, agglomerative hierarchical clustering. Okay. And the other is divisive clustering, where you are given n number of objects and you want to partition this full set of objects into two, so that they are maximally different from each other. Okay. So divide into two so that they are maximally divergent from each other, and do it recursively. So when you will divide it, for example, then you will get two groups, CDA, CDE and AB. And then CDE will get divided into C and DE. So this is recursive again, and eventually all will get divided into individual objects. Okay. So both these process results in a tree. So one is agglomerative from left to right, and the other is divisive from right to left. Okay, and here is an example of uh, agglomerative hierarchical clustering of genes. So you can see through this method, the genes have got clustered based on the gene expression pattern. Okay, based on the abundance of the RNA transcripts. And at the top, you can cluster genes, or you can cluster samples also, or both. Okay. So at the top, you have the clustering of the samples. So, and I will skip this actually, and quickly in 2-3 minutes, I will show you the RNA-seq pipeline that you can implement at Galaxy. And this was provided to me by my PhD student Ahmad. We should thank him for providing these slides. So this is an experiment done with Bacillus and Thracis strain volume, and RNA-seq data were collected when this strain was subjected to oxidative stress, okay, hydrogen peroxide stress. So, for example, to understand RNA-seq pipeline, we got the samples for wild type. Actually, I can I do this during the tutorial, right? Because this is that. So
Thoda explanation.
yeah, this will, okay.
So we break racks now. Okay. And now we get ready for the evaluation. So 1:30 time is there. We will run to that 1:30. We take this 45 minutes for exam.